no technical problems anymore, then let's get started. Yeah, so I'm very pleased to have Felix here. Well, he's a buddy from the MPI. So we did our PhD together some couple of years ago at the Max Planck Institute in Leipzig. Then afterwards, he was postdocing a bit in Jena and at the MIT. And then 5 years ago, he came back to Germany to another MPI, not the usual MPI for our series, but the MPI for infection biology in Berlin. And yeah, so he's working on ancient and present day pathogens and will tell us something about the evolutionary past of pathogens. Stage is yours.
Thanks, Martin. Thanks so much for the invitation. Meeting again like old friends and making some new ones along the way. To everybody, I hope you can hear me. For those who are online, otherwise, please drop a message. So yeah, today I want to talk about microbes. And microbes, as we all appreciate, colonize really every inch of our planet. And during this colonization, they divide and they acquire mutations, many mutations. If you, for instance, just take the bacteria that colonize in each and all of our bodies, what is called the human microbiome, we estimate that approximately 1 billion mutations are generated in our human microbiomes every day. And of course, that's an incubator for innovation. And we can use those mutations that are produced by bacteria in our microbiome or in any other context in order to understand something. For instance, we can use them to understand relationships of bacteria and can reconstruct transmission networks or translocation events. And sometimes the mutations that they acquire can also confer selective advantage. And by identifying the genes that are mutated, we can learn something about the most pressing challenges for the bacterial survival that obviously may affect like transmission, immune evasion, or resistance. And in my lab, we use computational genomics and like the lens of evolution in order to analyze those mutations and understand their role in the emergence, transmission, and adaptation of microbial pathogens. And we do that on dramatically different timescales. So maybe the reason why I'm here is because we do that on like a deep evolutionary timescale. So we use ancient DNA or ancient metagenomics of humans and non-human animals in order to understand the deep evolutionary history of those pathogens. We also utilize medical historical collections in order to understand really the arms race between pathogens and the host across the 20th century, which was a very important period in the development of our healthcare system, for instance, with the introduction of antibiotics, which has quite some interest. What are the changes that have been associated with that? And we are also working on rapid microbial evolution. So when I talked about the microbiome and all the changes that happen within there, we also try to understand the genetic mechanisms that really drive microbiome formation and what potentially can happen within our microbiomes and that makes us sick. And whereas for the first two, we use primarily just genomics, for the latter one, actually, we utilize usually native specimens, so specimens that we can culture, and then we sequence entire populations that we previously cultured in a petri dish. However, for the sake of the community that I was envisioning to meet here, I'm going to restrain myself today to only this part of the work we do. But of course, I'm more than happy to answer questions about the other things as well, if you have any. And for this prehistoric part, we are really trying to leverage ancient DNA to understand really the origins of human pathogens. And this interest is based upon like previous work I did, as Martin mentioned, doing my postdoc with Johannes Krause, where we worked on Salmonella enterica, and we discovered that the progenitor of a human-adapted Salmonella enterica lineage was identifiable in like farmers and pastoralist communities, but not so much in hunter-gatherers that we screened, which actually carried only a much more basal version of that Salmonella enterica progenitor lineage. And there's additional evidence that came out more recently from the group in Copenhagen who have like screened more general human ancient metagenomic data sets for pathogen presence, and they really identify an uptick approximately like 5,000 years ago, where we have doing the, what is, in general speaking, like the Bronze Age period considered, that we see that more infectious diseases seem to be present during that time. Now that may be because they actually increased in time, or that may be a sampling bias, because obviously there's a gigantic sampling bias in the ancient DNA field. That is not for certain, but what we can take from that is that certainly pathogens really seem to be present in the Bronze Age, and maybe that is also the time period when they have emerged. And that approaches like a more general question, what we know about the Bronze Age, that it was associated with those massive migration events where people from the Central Steppe moved into Western Europe, and people became sedentary, the population sizes increased, and very importantly, they introduced domesticated animals. And there is the theory out there that this introduction of domesticated animals really brought humans into more contact with many of the pathogens that then had the possibility for spillover and eventually becoming established in the human population. And when you take many of the modern genomes or modern pathogens and you try to molecular date them back in the past, many of them are kind of, have their most recent common answer in that period within, like, let's say, roughly the last 10,000 years with, of course, large confidence intervals. So now, if you think that domestication was important, then of course ancient DNA might provide like a unique opportunity to study those pathogens from their actual original reservoir in the animal context, which hasn't been done so far, because usually ancient pathogenomics is mostly, a byproduct of studying ancient humans. And that is one reason. The other reason, there are fewer specimens. And of course, there are particular challenges when you think about it that may be associated with identifying those pathogens in the animal record. And that is, first of all, that presumably those animals have rather been slaughtered when they were young and healthy, and because they were served as a food supply. So many of them did not have the chance to die of an infectious disease. Then, on top of it, they have been processed when they have been cooked, so they have been heated, which we know is further not good for their DNA preservation. And last but not least, in terms of how they have entered the archaeological record, that is also very different, because they've just been discarded, they've been in middens, and they've basically been kitchen waste instead of being buried properly and then potentially be excavated by archaeologists. So that all suggests that, finding pathogens in the animal record is actually rather slim. We nevertheless wanted to try it, and we tried it doing two different approaches. So in the first approach, we started to collect we generated our own collection of zooarchaeological specimens that we then wanted to sequence and search for pathogens inside. And in a second approach, we actually wanted to team up with other scientists who are actually working on the domestication processes of animals and just sift through their data and see whether we can find any pathogens in there. And I'm going to now present you two different projects, first on our own sampling, and then searching through metagenomic data. So as you all know, generating ancient DNA libraries from specimens, that is an expensive and time and heavy time investment. So we wanted to somehow find a way so how can we prioritize the specimens that are available for us in order to increase the chance of pathogen recovery. And what we came up with is to prioritize samples that show paleopathology, so in lesions that are indicative for infectious diseases. And therefore, we teamed up with Kamila Pawlowska, who is not just a zooarchaeologist, but also a paleopathologist, so really contributing expertise that certainly I did not have. So we could go through larger collections and identify specimens that show some levels, for instance, of periostitis, inflammation, or other signs and signatures that may be indicative for some level of infectious diseases. Of course, none of them being very specific, but at least give us some hinge and some leverage in order to prioritize specimen. And then we had access to specimen from the German Archaeological Institute, as well as from the lab of Philipp Stockhammer and people in Prague and some others. So we assembled a total of a bit over 300 samples from 34 different archaeological sites. Those sites ranged from Germany and Central Europe until Asia and Uzbekistan, Central Asia. So we have a vast geographic range. That is because we had not any prior information about where we want to look. We wanted to look as broadly as possible. We have also, as you can see on the bottom part of this plot, quite a substantial temporal range. So that is, again, because we didn't necessarily want to restrict ourselves to anything there. But we do have the majority of samples from this Bronze Age period based on pre-existing evidence that we think that period may be particularly important. And if we now look into like the composition of species that we have sampled, here you can see that on the right-hand side, different animal species. So we focused on domesticated animals, but if there has been like an interesting specimen from a wild animal, we also included that, even though in any way the differentiation is, as far as I understand, not always totally clear anyway. And then on the left-hand side, you can see that the distribution of lesional versus non-lesional samples. So for some sites, we had many samples with lesions. For some, we had very few. And we also included some teeth because we know from previous studies that teeth are particularly important for acute infections. So if you die from like a bloodstream type of infection, which of course in animals can happen as well, then we expect them to be in teeth and certainly not producing any lesions. Again, this was an approach to like really get like a big picture view about can we actually reconstruct pathogens from the animal record. So now we generated libraries for all of those, and then we sequenced them, 5 to 50 million reads, and then we screened them using a computational tool that I developed also together with my colleagues during my time in Jena. It's called HOPS. It's not particularly user-friendly. That's why I always encounter that many other labs don't use it, but I can only recommend using it because it combines identification and authentication in one go. We came up for the screening for like a set of rather, I would say, stringent criteria. So we expected at least of 50 reads to align to a taxonomic node of interest. And a taxonomic node would be, for instance, a species node, like a species level taxonomy identification of like a pathogen. And we had a long list of pathogens that we're interested in. Then we required some DNA damage to be present at the terminal ends of those alignments. Again, 10%. It is not extremely stringent, but I guess that is something that you could expect. Also, given that we have with 50 reads, of course, just a screening type of situation, so you don't expect too much damage to be necessarily present. We wanted a declining edit distance, which translates into the majority of your reads that you align to your taxonomic node. It aligns with a nearly perfect match without any mismatches. And then you have fewer reads that align with more mismatches and no read stacking. Because often in metagenomics, you see false positives because you align to some conservative region or conserved region in a genome, and then you have false positives because the read's actually from another species. So when we did all that and we sifted through our entire data set, we ended up with actually 15% or like 55 of our libraries producing hits to bacteria that are part of our list. So some of them are obligate pathogens. Some of them are obligate pathogens in humans, actually, thinking about zoonotic diseases. Those are here shown with those black arrows, whereas some others of the bacteria we looked at are also commensals, and they can lead to opportunistic infections in animals or in humans. And maybe one species here to point out is Salmonella enterica, which is a species that I used to be interested in the past, which can cause disease in humans ranging from gastroenteritis to systemic diseases, and has been implied in this Bronze Age period as well. And is clearly a zoonotic disease because still today it can infect many animals, as you may know. Then what we see, just trying to summarize this bigger data set, if you now look at the sample types down here, so you see we have bones with pathologies or teeth where pathogens were identified. We did not identify any signature in any of the bones that did not show a pathology. Confirming that overall the approach of targeting those lesional bones seems to be beneficial in terms of pathogen recovery. And in terms of sites, we can see that we have an overrepresentation of our the site from Uzbekistan called Tila Bulak, which only provide actually 30% of the samples, but 60% of the positive hits. That is suggesting either maybe at the Central Asian site there was a higher pathogenic load, even though we don't have necessarily any evidence for that, or because maybe the DNA preservation was better. So that is very hard to differentiate. While that, of course, is an interesting kind of, starting point to dig deeper into those pathogens. We didn't, for most of them, we don't have, actually for none of them, we have captures in my lab. So we couldn't necessarily sequence them on like an economic basis much deeper. So instead, but we nevertheless wanted to confirm them phylogenetically for their ancient authenticity. So we picked the 2 species where we actually identified quite a substantial amount of sequencing reads in our shotgun data sets. And we tried to, what I call like kind of project them into the phylogeny, those low coverage genomes. So what we do is we collect the present day genomic diversity of those bacteria and closely relatives of those bacteria. Then we identify the mutations that are phylogenetically informative of those species. And then we ask for those positions, okay, do we have any coverage in our ancient genomic data sets that at least allows us what is the branching point of those ancient genomes within the phylogeny. And I'm going to show you quickly the results for those two different species. So here you see for a species that we in our lab only call Rusio because none of us can actually pronounce the name. I'm not going to give it a try here. Where you see we had one genome, 0.5x breadth of 40%. So 40% of the genome has been covered at least once. And you can see it's located here. It's within the larger diversity of the species, infecting all kinds of different animals. But it interestingly falls together with 3 ancient genomes that have been previously reported from humans, suggesting that maybe there was a prehistoric level or lineage or diversity present at the time that so far has not been covered by what we know about that pathogen. It may have been of higher importance in the past, but of course that as many of those analyses requires further investigation, it's just the first peak, so I would not overemphasize it. And for the second pathogen, Streptococcus luciensis, this is, and I forgot to mention that Rusio is a common pathogen in animals today, particularly in cattle and swine. It's of economic importance. It's known to infect humans today as well. Similar is Strep luciensis, also known as a zoonotic disease, as well as a disease in a bunch of different domesticated animals. And here we had 3 genomes of various quality. And we see when we plug them into the larger diversity of Strep luciensis, which we had like 3 reference genomes here, you can see all 3 of them fall basal, but they do not fall basal to other closely related genomes like Infantarius and Galolyticus. And of course, a basal positioning in the species, but not in the genus, is reconfirming for its ancient position, given that it's, a couple of thousand years ago old. Okay, with that, I want to come to the conclusion of this first part, where I think we can show that indeed the zooarchaeological specimen can preserve DNA of pathogens, and that performing paleopathological investigations before can actually help you to inform specimens of interest. Of course, paleopathology is more for chronic disease than acute infections, so one always has to keep that in mind. And an approach that can allow us a phylogenetic placement from lowly covered genome, which is always interesting. Also, in a preliminary analysis of pathogens, because that's basically how we always start before we get a more deeply covered genome, if we manage to get it all. Okay. So let's get to the second approach, which is obviously much more economic. Just asking other people about sharing their data set. Of course, it comes with the caveat that other people need to share their data. So when I started my lab 5 years ago, I reached out to many people who work on zooarchaeological specimens, and despite many were very interested in the question overall, very few shared the data with me. One of them was Tina Warner. And Tina Warner at the time, or still is actually working on a particular site of the Sintashta culture, which is at Archaim, which is famous for the circular type of aerial view of the site. I mean, I didn't know it before, but maybe some of you have seen it before. So it's I want to tell you a little bit about the site. It's a fortified Bronze Age settlement. It was a pure pastoralist society, so subsistence is based on domesticated animals. It was a very innovative culture who like kind of picked up early on domesticated horses. They introduced chariots, and this technological kind of step allowed them to bring out their herds on like much larger pastures and rendered them overall fairly successful. And now Tina shared with me a data set of 23 ancient metagenomic data sets stemming from cattle and sheep that she was analyzing at the time. And we screened them again with hops, and we identified in one of those specimens a signature for Yersinia pestis, the causative agent of plague. And here just show you like one of those, how did like the initial signature look like. So here is what I mentioned before. That's an edit distance plot on the left-hand side, where you see on the x-axis the number of mismatches a read has, on the y-axis the number of reads. So you can see the majority of reads have no mismatches, and then it's declining and less number of reads with more mismatches pop up. And we see also some evidence of bad damage, as I said, that can be somewhat, a little bit unstructured given the low number of reads that we have in the screening situation. So that's a robust signature of Yersinia pestis. Some other pathogens are a little bit harder to identify, I need to say though. Yersinia pestis, I would argue, is one of the more easier ones. So of course, Yersinia pestis from a sheep, that sounded extremely interesting. We made a lot of investment. We performed capture that Tina had available and sequenced it deeper, and we managed to get it to a 2.3x Yersinia pestis genome with 75% breadth. That's not fantastic, but it's actually sufficient to do all the analysis that I'm going to show you. And of course, if you have any technical questions, I'm happy to answer them. However, before we go into the Yersinia pestis genome, let's quickly talk about the site itself. So Archaim, again, it's like here located in the middle in Russia. The sample is a sheep tooth. And interestingly, here on this map, you see all the other prehistoric Yersinia pestis genomes that have been reported. You can see they're spreading across all Eurasia. And there is one site, it's WISE 386, just to the west of the Archaim sample. That is also a sample, it's a human, all of them are human samples, by the way. So this is the WISE 386 is another Sintashta individual believed to be in like the same temporal period as well. And there has been identified with a Yersinia pestis positive signature. It has never been phylogenetic placed though, because it's also fairly low coverage. So that about the data set. And then obviously the first question was, is it the sheep actually a domesticated sheep, or is it maybe also a wild sheep? So we performed the PCA analysis using including different sheeps, wild and domesticated sheeps, and you can see ARCO17 was exactly on top of the domesticated sheep bubble, confirming that we here reconstruct the first Yersinia pestis genome from a 4,000-year-old domesticated sheep. So how does this Yersinia pestis genome now relate to what we know about Yersinia pestis? So here is a phylogeny that includes all modern as well as ancient Yersinia pestis. So on the top, there are many like mostly modern diversity. So it's today a fairly diverse pathogen species. And down here in the bottom in blue is shown what we call the Bronze Age lineage, or the late Neolithic Bronze Age lineage. And our sheep genome actually falls directly into that lineage. And this lineage previously has exclusively been identified from humans that are dating back between, I think, 3,000 to 5,000 years ago, and seemingly is extinct today. So at least no representative in modern times has been found so far. And when we now zoom into that lineage, you can see our sheep genome is just, so you, first of all, you see obviously the phylogeny, so a two-dimensional representation of relationships, and on the x-axis, you see the age of the different specimens, the carbon dates. And you can see the sheep genome is up here, and it's most closely related with this RISE 386 genome, the one that I mentioned that was just west of the Archaim site, so which is culture-wise most closely related, like the humans were most closely related in terms of culture, and also the pestis genome, so the infections they received, the sheep as well as the humans, they're also most closely related, showing that the strain that is infecting the sheep could also infect humans. And what is very peculiar about this lineage, which, I mean, just by looking at the overall by pestis diversity, which is, as I said, fairly diverse, this Bronze Age lineage is actually like pearls on a string going through time. That is a pattern that you would not necessarily expect by chance. It's what we call a ladder-like phylogeny. And in our first now like deeper investigation, we wanted to ask the question, so did molecular evolution potentially contribute to this peculiar shape of this lineage? And therefore, I need to make a short excursion into theory. So if you have a population that evolves under neutrality, then it just randomly accumulates mutation, and the phylogeny looks somewhat like a star. This is not what we observe. We instead observe like a repeated reduction of diversity. That can happen through bottlenecks, so when just like randomly genetic variability is again and again removed from the bacterial population. That can lead to the pattern that we observe, or of course, natural selection, where either positive selection or adaptive genotypes outcompete not so adapted genotypes and replace them, or purifying selection, preventing from additional diversity to accumulate. Now, how do we differentiate between those different models? We take advantage of the fact that genetic mutations can come like into two types of flavors. They can either be non-synonymous mutations, so mutations that change the amino acid code, and we generally believe that those non-synonymous mutations are a hallmark like of positive selection. So you have something that's different, and then that sweeps through a population. So we expect an excess of non-synonymous mutations under positive selection, whereas under purifying selection, we do not want any functional changes because all of them are bad. And so they are removed, and we have an excess of so-called silent mutation or synonymous mutations. And of course, under a bottleneck scenario, which means random removal, we expect them to be in some kind of equilibrium. Now, to test that, we use a classical test. It's called a ratio test. It's a DNDS test. And here I just show you the results where we contrast the extant diversity, so all the modern diversity, versus this Bronze Age lineage. And you can see extant diversity, as has been previously shown, evolves under neutrality overall, because the ratio is approximately 1, or at least the confidence intervals are across 1. And our Bronze Age lineages instead is actually having a DNDS ratio smaller than 1, suggesting it evolves overall under purifying selection, contributing to this ladder-like phylogeny. Contributing means it's probably not the sole cause of it. Yeah, bottlenecks and so on is obviously in the pathogen life cycle always an issue, and most likely further contributes to it, but more difficult to show. Now, how can we now actually use this sheep genome in order to learn something about the transmission cycle of pestis, and ideally the transmission cycle of Yersinia pestis of this ancient lineage in the deep past. So therefore, I want to briefly talk about, okay, what do we know from the Yersinia pestis life cycle or epidemiology today? So Yersinia pestis is maintained in flea populations, and those fleas live on rodents, like marmots, for instance. And then only occasionally from those fleas on rodents, occasionally there's a spillover into humans, as well as sometimes also known into other animals, including domesticated animals. Now, however, one specific aspect of this ancient Bronze Age lineage, and that also is true for the sheep genome, is that it lacks actually genes that are implied in efficient flea transmission. So we think that fleas may not play a role as important for the transmission of that Bronze Age lineage as it does today, arguing there may be other mechanisms that be more in the foreground of the transmission cycle. Of course, there's human-to-human transmission, as we've seen, for instance, in the Black Death in the Middle Ages. But that, again, is unlikely to be the driving factor in the Bronze Age because populations were just too small. The distances the pathogen would have to travel are too enormous because, I mean, I haven't talked about that, but you see this ladder-like phylogeny, but there's no geographic structure associated with it. It goes back and forth across Eurasia. You know, Yersinia pestis usually kills human individuals within 1 week, so that doesn't seem to be a feasible scenario. So we are a little bit in the dark, but let's assume the ecology of the pathogen has not changed so dramatically from the Bronze Age to today. When we look at the phylogeny from that point of view, then we would believe that probably the human infections that we observe, they are mostly spillover events. We don't think it's maintained in humans. I think there's very little evidence for that. And that means we can separate this phylogeny into 2 different evolutionary scenarios. So we have this backbone here, and that's evolution that must happen within the reservoir, whatever the reservoir may be. And then we have all those singletons here, all those little humans that we happen to sample because we're interested in human demographics and started sequencing ancient individuals from teeth. But it's just coincidence, and it's actually all are just individual spillover events that may or may not have been associated with some small-scale outbreaks of the pathogen, you know, which is in theory very much in line with our observations, because those people that are, or like this individuals that are positive for pestis with this Bronze Age lineage, they normal burials, no mass burial, no like pandemic type of scenario associated with them. So we think those are more sporadic events. And now when we believe that we have all those humans that is like independent spillovers into humans, that means you can consider when I coming back to the beginning, that this is a repeated introduction into a new environment, which is the human host, where it may have survived for some time, may have infected multiple individuals. That's all possible. And that obviously recreates an evolutionary pressure. And this evolutionary pressure is repeated over thousands of years, multiple times. And that evolutionary pressure may result into certain genes being mutated because mutations in those genes provide an advantage, which is something that we call parallel evolution. And we can test whether parallel evolution is happening by asking, okay, if we take only the mutations that fall into those, that are presumably happening not within the backbone, but during those human infections, do we observe more genes to be mutated multiple times by chance than what we expect? And we can do very simple simulations. We just take, okay, we observe 100 mutations and no singletons, and just, okay, sprinkle them randomly over the genome and ask, okay, how many of the genes are actually mutated multiple times? We do that, 10,000 times, and we get a distribution, and we can ask, what's our observation? And when we do that, we actually identify on those external data and branches that we have an increased number of genes with 2 or more mutations than what we would expect. And many of those genes seemingly are associated with virulence, even though the knowledge about those genes is very limited. So it's not crystal clear. There's not, at least we have not identified necessarily a smoking gun where we put our hand in the fire and say, we think, that may have been something that's super important and should be investigated. Of course, Yersinia pestis is an S3 pathogen, so it's not so easy to work with that. But very reassuringly, we do not see a similar pattern when we just do the very same for the backbone. So here where we assume, okay, to the reservoir, it must have been adapted already, and it survives within that reservoir. So there we do not see evidence for parallel evolution. Only in those dead-end branches, arguing in favor of the hypothesis humans, and most likely sheep as well, were not the reservoir, because sheep is contributing to the pattern we observe on the top right-hand side plot. But instead, they were just accidental spillovers into the human population. And we can take all that information and try to coax it into like into a scenario that we think may have happened in the past. So we have this reservoir, presumably wild animals. We don't know what it is. And from that reservoir, it can either, of course, infect directly humans, or it can cross over livestock. And then livestock infects humans. And we think that this route actually may have been in place more generally, because as I mentioned in the beginning, those pastoralist societies, huge herds. So those sheep herds, and as I said, also other domesticates, they had a much higher chance of encountering somewhere in the wild a diseased animal that may have just died there and is laying somewhere on the pasture, and they're just eating grass. And we know from today that sheep can get infected, for instance, by licking diseased marmosets. And then you have this huge flock basically collecting pathogens in the wild, and then you bring them home, and then you slaughter them, and then you have like a bloodborne transmission event while slaughtering it, and then you become sick. And that is also something that has been described today. And that this path of infection, at least of humans, may have been the problem, or the path that has occurred more often than maybe a flea-based transmission, given the genetics of the pathogen. And of course, that is some level of speculation. Summing it up, I hope I could show you that we have like this 4,000-year-old Yersinia pestis genome from a domesticated sheep. Sheep and humans are part of the very same single lineage. Natural selection differentiates Bronze Age and extant by pestis diversity, and that sheep, as well as potentially other domesticates, has elevated the infection risk of humans during the Bronze Age. And again, also that paper is out on bioRxiv. Happy to have you have a read. And with that, I want to thank, of course, you for listening. I want to thank my team. That is work I presented today was from Ian Light, one of my first PhD students, and Elke Runge, a postdoc in my lab, and of course the many collaborators. And yeah, no, again, thank you for listening. And I'm happy to take questions.